Thursday, looking at the blood supply to the heart. We had 6 arteries and 3 cardiac veins. And so some of them are found with 2, some are only found with 1. So what artery or arteries is the middle cardiac vein found with? It's found with 1, it's marked 1. If it's found with 2 or more, then mark whatever is applicable. Want more time on number 1? Question number 2. So we're starting off the physiology part of the heart. Well, we're doing the physiology part of the heart this week except for blood pressure. We're going to do some review of action potentials in neurons and muscle cells. So as you look at this action potential along the axonema of a neuron, right, which of these channels here create that membrane potential? Now, leave that question up as we start our review. So if you don't remember this, you might want to go back and review the chapter of membrane potentials, both for muscle cells and for neurons. Okay? I don't remember the number, but for an area that they start really early with membrane, starts talking about membrane potentials, and it's also reviewed again with the muscle and the neuron itself. All right, so putting that diagram up there. So this would be along the neuron, all right? And that's the axonema, although sometimes we see it with that as well, all right? So you got to remember from Bio 430 what this potential is called. Resting membrane potential, all right? And this one would be called graded potential. And this point right here would be called types of potentials in muscle cells because they're both considered to be irritable. Hi, we're having lecture in there, so do you mind if you guys sit? All right, so anybody remember what do we call this one? Threshold. And then that one would be,
Depolarization?
pardon?
Depolarization?
Yes, it is, depolarization and repolarization, but as a potential, what do we call it?
Action.
Action potential, yeah. Looks like nobody gave lecture in here, this board is sparkling clean. All right, so, what do we mean by a potential? What is it? What are we talking about here? Not all cells have potential. When they do, they're considered to be irritable, right? So, nerve tissue, nerve cells, and muscle cells have action potentials. Are irritable. They're also conductive. They can conduct that irritability, and the muscle cells, we talk about them having another characteristic, which is contractibility. All right, so both muscle cells and neurons are irritable and have conduction. So when we're talking about a potential, we're talking about a difference in charges on the inside of the cell and the outside of the cell, right? So if this is the cell membrane, and this is outside the cell, and that's inside the cell, do we have fewer or more positive charges inside the cell? The inside the cell relationship to the outside of the cell, is it considered to be have fewer positive or more positive? No, fewer. So we write it in this fashion. This would be the plasma membrane of a neuron or a myofiber. Okay, so the inside of the cell has fewer positive charges, not necessarily more negative, but definitely fewer positive charges, and the outside has more positive charges in comparison. That's what creates a potential, all right? And that potential is expressed in electrical current. So if available, if we open up channels, which way are the positive charges going to move? We have more positive charges outside than we have inside. The positive charges are going to move from outside the cell to inside the cell. That flow of charged particles creates an electrical current. That's the definition of electricity, is the movement of charged particles, and that's by diffusion, okay? So they go from their area of high concentration to their area of low concentration. We have more positive ions outside the cell, so when available, when we open up ligand-gated channels, or we open up voltage-gated channels, the positive ions move into the cell, okay? So, do you remember the three major reasons why in neurons and muscle cells and myofibers we have more positive cells outside than we have inside? While you're thinking of that, I'm going to move this diagram over here so I have more accessibility. So we have two of those reasons have to do with moving ions across the membrane and how we move them in a relative fashion, all right? So let me redraw this plasma membrane. Again, plasma membrane of a neuron or a muscle cell. Last semester we talked about skeletal muscle, okay? And neurons. We're doing this in preparation to talk about cardiac muscle, but it's still a muscle cell. We have our plasma membrane, and what ion is higher number outside the cell than inside the cell? Sodium. So if we diagram sodium ions, we're going to have more sodium ions outside the cell than we have inside the cell, okay? What about potassium ions? Where are they highest in number? Inside the cell, okay? So potassium ions are going to be more prevalent inside the cell. Now, for a cell that doesn't have a membrane potential, those charges balance out. Okay, that's why they're not irritable, because they have an equilibrium in the number of positive charges inside and outside the cell. Now, how can we get fewer positive ions inside the cell? Not quite, we're talking about resting membrane potential. Okay? So how do we get fewer positive charges? If we're starting out with this, how can we get fewer positive charges inside the cell? Leak channels, and if we want that potassium to leave, then we have to have a leak channel for potassium, right? So we have leak channels for potassium. Leak channels mean that they are always open, okay? They're not gated. So one of the reasons that we have a resting membrane potential are the leak channels for potassium. But we also have leak channels for sodium. So potassium is going to go out, all right? We also have leak channels for sodium. So sodium is in higher concentration outside the cell. So if we have leak channels for sodium, sodium is going to come into the cell. And how does that contribute to having more positive ions outside the cell? We're still at equilibrium, okay? We're just exchanging. Sodium's going to move down its concentration gradient, potassium's going to move down its concentration gradient until both ions are in equilibrium inside and outside the cell. And that doesn't give us any higher number in any different locations. So what do we have to do in addition to this, adding leak channels? We will get to that, but we need to modify this first, okay? Pardon?
Sodium potassium pumps?
That's what Joyce said, but we'll get to that. That is another reason. But before we leave this, how do we change this from being in equilibrium? Because if all we have is what I have on the board, we're going to reach equilibrium. And we'll have the same number of sodium and potassium outside the cell as inside the cell, and we'll still have a balance of positive ions.
There are much more leak
Yes,
channels
there are for potassium.
Okay.
So we have more leak channels for potassium than we do for sodium. And so our way of phrasing that is the cell is more permeable for potassium than it is permeable to sodium. And that permeability is due to an increased number of leak channels. All right? So to add to this, we have more leak channels for potassium, and we have fewer leak channels for sodium. So the cell is more permeable to potassium, which is going out, than it is to sodium, which is coming in. So this means we have a net loss of positive ions, okay? So that's one of the three reasons for why we have fewer positive ions inside the cell than we have outside the cell. And then a couple of you mentioned the sodium potassium pump. So I need a little bit more room in my plasma membrane here. And I'll just draw one, but there are several. So in the sodium potassium pump, it moves both types of ions, sodium and potassium. When unlike our leak channels, it requires ATP. But how do we get our unequilibrium in the movement of positive ions?
2 to 1.
2 to 3, with a difference of 1. So remember, we're going to lose more positive. We're trying to return this back to where it was. Remember, we need energy, so we need to go from low to high instead of high to low. Low to high would be sodium out and potassium in, right? In both cases, those are moving from low to high, because we're low in sodium inside, high in sodium outside. Low in potassium outside, high in potassium inside. So both of them are going up their gradient. So we're going to move potassium into the cell. And we're gonna move sodium out of the cell. And we contribute to the potential by the numbers. So we lose 3 positive ions of sodium for every 2 positive ions of potassium, with a net loss of 1 positive ion. So we're losing positive ions again, all right? So another reason why we have fewer positive ions inside the cell than outside the cell is our sodium-potassium pump. With a net loss of 1 positive ion. So that's the second reason why we have fewer positive ions inside the cell than we do outside the cell, all right? Now the third reason has to do with something that doesn't cross the plasma membrane, and that's because of its size. So inside the cell, we have large proteins that are too big to cross the plasma membrane, and they have a net negative charge. Okay. So that contributes negative ions to the inside of the cell. So that's the third reason why the inside of our cell is more negative than the outside. All right? So I want you to be able to recognize potentials and the channels that we associate with them. So with the resting membrane potential, we have 2 channels. We have leak channels, sodium and potassium, and we have the sodium-potassium pump, which is also a channel. Okay? And that's what helps to create the resting membrane potential. Okay? So by now, you should be able to figure out the answer to question 2. So may I erase the left half of the board? Let's have that down. And we'll look at our graded potential. Okay? So the cell that's going to be irritable requires a resting membrane potential, because if it was in equilibrium and we attached acetylcholine or norepinephrine and opened up a ligand-gated channel, nothing would happen, because the ions would be balanced on either side, and then there wouldn't be any difference. Okay? It'd be just like opening your mouth and not moving your breathing muscles. No air doesn't move in or out. So we have to create some purpose, some force, that concentration gradient, to move ions into or out of the cell to demonstrate that irritability. All right, so that's our resting membrane potential. And then we have the ability to either increase or decrease the number of positive ions inside the cell. So if we look at that graph, and this is positive ions up here, and we typically talk about the resting membrane potential at about 70 millivolts, between 55 and 70. That referred to the number of ions that had to cross the membrane to bring the inside of the cell and the outside of the cell in equilibrium. Okay? So for a neuron or muscle cell to have a resting membrane potential of -70, that meant that the minus means that positive ions have to flow into the cell to make the negative inside the cell more positive. And enough positive ions have to flow into the cell that they create a current of 70 millivolts. The minus means they have to flow into the cell. If it was a plus, it would mean they had to flow out of the cell. Right? So that's what we mean by measuring that current. potential, and then we can add positive ions, or we can lose negative, all right? Typically we add positive. And what do we call this movement? When the inside of the cell becomes more positive? Not, well, it is a graded potential, but what do we call the inside of the cell becoming more positive? Depolarization, okay? So this is depolarization. And it can occur when we add positive ions, typically sodium, but it could be calcium as well, okay? Or sometimes magnesium. Or seldom, it can occur when we lose negative ions. It's more common to add negative ions. Then if we have a situation where the inside of the cell becomes more negative, that's called hyperpolarization. Or if we start from a depolarization and we become more negative, repolarization, okay? So this is hyperpolarization. And this is repolarization. And that can happen by a loss of positive ions, and that's usually potassium. Okay? So when potassium leaves the cell, the inside of the cell is becoming more negative, or a gain of negative ions, and that's usually chloride. When chloride comes into the cell, it's becoming more negative. All right? So, somebody jumped the gun on me. What kind of potential is this? A graded potential, okay? So what types of channels in the membrane cause graded potential? What do we normally do? What normally causes, if we're talking about skeletal muscle cell, what normally causes positive ions to come into the skeletal muscle cell? Ligand-gated channels, and exactly right, and the ligand would be acetylcholine. So when acetylcholine binds to the membrane receptor on the skeletal muscle, that's an ion channel, and it opens, and sodium ions come in. Same thing between a preganglionic and a postganglionic neuron. It doesn't matter whether it's sympathetic or parasympathetic. The preganglionic neuron releases acetylcholine. It binds to a receptor on the surface of the cell body of the postganglionic neuron, and that opens a sodium ion channel. Okay? And so that becomes a graded potential. Now, graded potentials, as you can see here, can go either direction. They can make the inside of the cell more positive, or they can make the inside of the cell more negative. Okay? And they do so because of a chemical binding to a receptor. So depending on what that receptor is, whether it's open to positive ions or to negative ions, will depend on whether our graded potential is a loss or a gain of those charges. Now, one of the reasons we call it graded is because if we have, and I'm going to erase this here if I may, and diagram a neuromuscular junction at a skeletal muscle. So when we look at that, if you look at a lower motor neuron, synaptic gluton, releasing acetylcholine. And acetylcholine diffuses across the synaptic cleft and binds to receptors. That allows sodium ions to come into the cell. So let's say that I'm trying to pick up this box. And I try to pick up the box, and I try to pick up the box, and it's not moving. What do I need to do to get my muscles to contract more strongly and create a greater force to pick up that box. I have to fire more often or fire more neurons so that I get more acetylcholine. We talked about spatial summation and temporal summation. So the more acetylcholine I get, the more channels I open. And if this comes off, another one's going to be right there to take its place, so that same channel is going to stay open longer. All right, so that's what we mean by graded. We can make it allow more ions in or fewer ions out, or vice versa. And so we talked, illustrated this with throwing a rock into a pond or a lake, and it creates waves. And if it's a small rock, those waves aren't going to go very far before they dissipate out. They lose their energy. If it's a great big boulder, the waves are higher in amplitude and they travel farther across the pond, maybe all the way to the other side. That's what we mean by graded. So the current coming in or the current going out can be weaker or stronger. All right, it can travel farther along the cell body or the muscle cell, or not as far. So all of those are graded potential. So the channel that we associate with the graded potential is a ligand gate, which means ligand is chemical. So it's some kind of a molecule binding to a protein, opening up a channel for the ion to cross when normally it would be closed. That's the difference between a leak channel and a ligand and any kind of gated channel. The leak channels are always open, and the amount of diffusion across depends on concentration gradient. So these are not always open, all right? And the amount that diffuses across depends on how long they're open and how many of them are open, as well as concentration gradient. Okay. All right, so in this diagram here, number 2, which of those channels would result in the difference between the resting membrane potential and that dotted line, the red line? So since I have a muscle cell up here, I'll stick to it for our illustration. Anybody remember what we call this membrane of the muscle cell? We have 2 root words for muscle. One was myo, what was the other? Sarco. So a sarcolemma, all right? And our ligand-gated channels were in this motor end plate of the sarcolemma. And as we build up a number of positive ions, we're increasing their concentration, right? And so if they're high in concentration here, but not high in concentration there, those positive ions are going to diffuse away from the motor end plate and out under the regular sarcolemma. They're going to move up here, away from the motor end plate. And we have a different type of channel here. It's still gated, but it's the current that's going to open it. It's the number of positive ions flowing by those protein channels that will open it. So we have both sodium and potassium channels. And if sufficient numbers of positive ions flow by them, they open, all right? So these are identified as voltage gated channels. So for those first channels, what's going to open them? What's going to cause them to open? It's the amount of our graded potential, right? If our graded potential reaches the minimum current to open these, they open. If it doesn't, they don't. So what do we use as the term for that minimum current? Threshold potential. in this case, they're gonna open because of the ligand-gated channels, enough of them being open. And since they're ligand-gated, that may not always be of sufficient voltage strength to open it. But as we move farther down, and we have another set of voltage-gated channels, the current of our graded potential is not going to be strong enough to open these because they're too far away from the motor input. So what's going to open these voltage-gated channels? This is called self-propagated or saltatory conduction, because then these open, enough positive sodium ions come in that will travel a short distance, and that will be sufficient. Those sodium ions coming in here will be sufficient to reach the threshold potential for these. So these voltage-gated channels are going to open because of an action potential, all right? So the threshold potential can be reached either because of the graded potential being high enough, or farther down the muscle cell or the axon, it will open because of an action potential. And the action potentials are always strong enough to open voltage-gated channels. Okay, that's the way they're set up. Which brings us to our fourth potential that we wrote on the board earlier, and that is the action potential. So what kind of channel is going to create an action potential then? Voltage gated, okay? And typically sodium and potassium are utilized. VG stands for voltage gated. But we also have calcium voltage gated channels, as we saw in the sarcoplasmic reticulum. And in the synaptic bulb, we have voltage-gated calcium channels there. All right, that was a quick review over what took 2 days to cover last semester. And again, if this seems a little bit strange or you don't remember talking about it, then go back to the previous chapters on neuron and muscle. And Mary Beaver talks about it in her chapter on plasma membrane. And you can read about those receptors there, okay? Different types of channels. Any questions on this before we move on and tie this information into the two different types of cells we see in the heart? Okay, so in other words, if we were talking about the, can I erase this? No, I'm just going to redraw it, but as a neuron instead of the cell body. And then I'll come back to this. So if we're looking at the cell body of a neuron, and then its axon, and we'll just not draw in the dendrites and so on, okay? Here, along the cell body or the dendrites, we have ligand-gated channels that require a chemical to bind to it. So that creates a graded potential. As positive ions, typically sodium, move into the cell, we may not have enough to reach the current required here by our voltage-gated channels. Okay? In fact, we may get negative ions coming in, chloride ions coming in. But if we have enough, then that do not, we're going to lose some through the fusion. But if we have enough coming here, then that current flowing by our voltage-gated sodium channels will be enough to open them, okay? However, down here, we also have voltage-gated sodium channels. That current coming from the cell body is insufficient to go all the way down the axon to open voltage-gated channels all the way down the axon. That's just sufficient to open the ones that we have here, and these are actually have a higher sensitivity, they're open with a lower number of positive ions. So when this opens, because of the graded potential, these are still voltage gated, but it's opening because of the graded potential being strong enough. This is going to allow positive ions to come into the cell. And those will travel down the axon, and that will be sufficient to open up the next set of voltage gated. And that's what's going to continue it to open. So we talked about dominoes. And so that first domino gets pushed by your hand. But then after that, each domino falling over is enough to knock the next one. You don't have to keep pushing the next domino over, okay? So the first voltage-gated channel to open opens because of a graded ligand-gated potential. And then the rest of them are going to open because of the action potential opening the sodium channels prior, just proximal to them. Okay? All right. So looking at the heart, and we'll look at this again on Thursday when we actually talk about an ECG, we have a region, several regions of specialized muscle cells that are not contractile. Okay? They have lost most of their actin and myosin and have taken up glycogen as a source of energy, and they are considered to be autorhythmic. So the two types of cardiac cells that we're going to talk about are autorhythmic and contractile. And the contractile cells are very similar, although not identical, to our skeletal muscle cells. What does autorhythmic mean? It means they don't require ligand-gated channels. Okay? They don't require a neuron releasing a chemical binding to a receptor on their cell surface. They have them, but they don't require them. This is why you can cut the heart out of a frog, or cut the heart out of an individual who is on life support, but otherwise brain dead. And that heart, if it's not, temperature has not been decreased, and it should be happens in transport, so it's not building up waste products, right? Because there's certainly no circulation moving through the heart. So they usually chill it, so that it's not going to be contracting. But it will contract until once it's removed from the body. Definitely no neurons are connected to it anymore. And typically when it's placed in someone else's chest and the large vessels, superior vena cava and pulmonary trunk and aorta and inferior vena cava are sutured back in place, large vessels from the recipient heart, a recipient individual, and that heart starts to beat again, nerves are typically not attached. Okay? And yet the heart will beat. So these autorhythmic cells create their own depolarization without a ligand-gated channel being required. Now, individuals that have heart transplants then have special requirements or special relationships to exercise. About 20 years ago, a young woman in Southern California, in her late 20s, early 30s, developed a viral infection of her heart, which resulted in cardiomyopathy. Weak heart muscle got larger, and it wasn't efficiently moving blood through her body, and she required a heart transplant. Prior to the illness, she was in the process of climbing the highest mountain on every continent. And as she was flying to the hospital for the transplant surgery, she looked out her window and saw the Matterhorn at Disneyland. And she made a vow that she was going to keep doing that, even though that was a toy mountain. So after surgery, she started back in her exercise regimen. So for the rest of us, as we exercise, before we even have a change in oxygen supply, just the movement of our joints, there are receptors from our joints, proprioceptors, that go to the brain, all right, and trigger activation of the sympathetic nervous system. And our heart rate starts to increase before we actually really need that increased movement of blood supply through the body. She wouldn't have had the sympathetic nerves, accelerator nerves, attached to her SA node and AV node. So how did she get her norepinephrine to increase her heart rate? What's the other source of norepinephrine in the body besides the sympathetic nervous system? Endocrine system. And what organ? It's called epinephrine instead of norepinephrine, but it's chemically almost identical. Anybody want to help her out? Another name for epinephrine is adrenaline. So that would be the adrenal glands, okay? And we talked about this, but we talked about the similarities between epinephrine, between endocrine and nervous system. So her heart rate would take a while to increase, all right? But then it would be increased. She would have more oxygen moving through her body. And then when she stops to rest, our heart rate normally drops because of acetylcholine. Do we have endocrine organs that release acetylcholine? The only source of acetylcholine is the vagus nerve coming into the chest, all right, or the pelvic splanchnic nerves down in the pelvic organs. So there's no endocrine organ to stimulate to balance out the increased heart rate due to the norepinephrine. So the only way to slow her heart down is to wait for the epinephrine to be removed by the kidneys, by the liver, which takes almost 30 minutes to an hour and a half, depending on how long it is. So her heart rate's not going to come back to normal within 5 minutes, like it does for most of us, because there is no endocrine acetylcholine. You can get the norepinephrine through the adrenal gland, but not any kind of acetylcholine. Okay? So there are some factors in there that we lose when we don't have the ability to modify the heart rate. But that's what the purpose of the sympathetic and parasympathetic nervous system are to the heart, is not to trigger heart rate, but to modify it. Either raise it or to lower it. So these cells, as I indicated, are identified as autorhythmic cells. And they are modified muscle cells that have lost most, if not all, of their actin and myosin. They're found primarily in these two locations. Okay? So the SA node, known as the pacemaker of the heart, because it typically creates the most numerous action potentials, unless it's not functioning. And SA stands for sinoatrial. So if you're identifying this on a lab quiz, you need to write the whole thing out. So that's what SA node stands for. And then we have the AV node, which is atrioventricular. So how do you think the current, the electrical current flow of positive ions created by the sinoatrial node gets to the atrioventricular node? It's passing through the contractile muscle cells of the heart. And the night before we leave, we'll talk about those contractile cells. Do you remember a type of junction that creates a communicating gap between cells? Good. Yeah, gap junction. So one of the types of junctions that connect muscle cell to muscle cell are gap junctions. And so that current flows from cell to cell, all right? Triggering each subsequent cell to contract. Now, and we'll review this on Thursday. Why don't we continue to have these arrows moving down through the ventricles? Pardon? So the atrial contract, but why don't we have that contraction sweeping all the way down to the ventricles? Why do we stop it right here? So you can see the arrows aren't continuing. Why do they need to? They do. So the cycle of contraction of the heart, that we'll talk about on Thursday is the heart's at rest, 2 atria contract, and then the 2 ventricles contract, and the heart's at rest. And then the 2 atria contract, and the 2 ventricles contract. Why don't we just do this? Where's the blood going from? Do we have any outflow down at the apex of the heart? No, our outflow is up here in the pulmonary trunk and the aorta. So we have to stop this and then bring it down and restart it from the apex. So what stops this is something called the cardiac skeleton. It's a ring, if you will, a band, a couple of rings of dense irregular connective tissue, which is not conductive. It forms the valves, the atrioventricular valves, and it forms the tissue around the valves to which the valves anchor. And so as the ions are traveling through the cardiac muscle cells, they reach the cardiac skeleton and they're blocked. So they typically, though, will activate the atrioventricular node, and then that will carry the current down to the apex through another set of conductive cells known as the bundle, the atrioventricular bundle, or bundle of His is the old name. We're not being gender specific. It's not his and hers. His is somebody's name. All right, so that crosses from the atrial wall here into the interventricular septum. So that's the atrioventricular bundle, or bundle of His. And then 2 channels will travel down the interventricular septum, and those are known as bundle branches. That takes them down to the apex of the heart. However, off of the right bundle branch, we have the moderator band. And that takes it over to one of the right papillary muscles. Remember I said that was a special conductive tissue? At the apex, then it goes up the outside, so not just 2 of these, but the entire outer wall of the heart. Looks like just 2 of these sets, but it's going 3-dimensionally all the way around the ventricles. Now these are called conduction myofibers, or the older name is Purkinje fiber. And these are right past through the outer ventricular wall. So the reason the heart, the SA node, the sinoatrial node, is called the pacemaker is because without influence from the vagus nerve, this is going to fire action potentials at about 100 times a minute. Your normal heart rate is somewhere between 65 to 72 for most people. Mine's around 78. My son's is around 48. So there's other factors like exercise that come into play. Okay? The AV node is a little bit slower. That's about 60 times a minute. So if the SA node is healthy, it's going to essentially pull the rest of the heart. The AV node is only firing 60 times a minute, and the SA node fires 100 times. Then this is going to get to the AV node before it has a chance to fire again. So it's going to kind of govern that. The bundle and the bundle branches are basically about 40 times. And individual muscle cells do about 15 times a minute. Okay? So that each contractile cell will also be polarized on its own, but too slow to really keep you alive. And that's what happens with defibrillating hearts. Each cell is just kind of contracting and firing on its own without any coordination. It's not moving any blood through the heart. So this is what creates that electrical current that when we compare to the ions in the skin, we can measure as an ECG. So that's what we'll talk about on Thursday. Right now, I want to look at what's going on inside these autorhythmic cells. erase the right side of the board and everything but the word autorhythmic cell. You guys are ready with that. So may I erase this? You guys have that down? Okay. So if we measure the change in current at the autorhythmic cells, they typically get about minus 60 millivolts. So to get them to equilibrium, it takes a number of positive ions moving into the cell to create a current of 60 millivolts. All right, this is a little bit in between our neurons and muscle cells. And you'll notice that we have something missing here. So what is missing in our diagram here? Exactly right. No resting membrane potential. So we don't have any resting membrane potential in the autorhythmic cells. We don't have any ligand-gated channels to kick them up or below that resting membrane potential. They're constantly either depolarizing or repolarizing. That's what we mean by autorhythmic. All right? So on their own, either losing or gaining positive ions. So they have some kind of channel that allows that difference in concentration gradient to affect it. So the red line represents potassium ions. Okay. The black line represents calcium ions. And the blue, you see those things where you write the word green, but you write it in a blue color? You try to confuse people that way. I'll try not to do that. So all of this is depolarization, right? We're adding positive ions to the cell, and then we repolarize through the loss of potassium ions. And that brings us down to a minus 60. As soon as we reach about a minus 60 millivolts, we start the process of depolarization again, okay? So looking at that, the channels that open at minus 60 are identified as funny channels. That's actually what they're called, okay? They're typically primarily for sodium ions. Although early on, potassium is moving through them too. But as far as we're concerned, we're going to consider them to be sodium ion channels. All right? The reason they're called funny is because they open for two different reasons. Remember I said they open when the inside of the cell reaches a negative 60? Is that a voltage or is that a chemical? The voltage. So these are voltage-gated channels. Only instead of becoming open when the inside of the cell reaches a certain positive level, they open when the inside of the cell becomes to a certain negative level. So these are voltage-gated channels. cell gets to that minus 60, a molecule will attach to many of these. Not all of them, but to many of them, causing them to open as well. And this is a molecule known as cyclic AMP. So they're also ligand gated, but from chemicals on the inside of the cell instead of on the outside of the cell. Okay? So these are,
What is the CAM doing?
it is binding to the funny channels and causing them to open. Open a little earlier than they might, but also just causing them to open. So because when we use norepinephrine, more cyclic AMP will bind to them and we'll see them open even earlier than they normally do. So that's, those two reasons are why they're called funny. They're both voltage gated and ligand gated, and the ligand is inside the cell instead of outside the cell. So it's binding to the inside of the cell membrane, all right? Then, when they reach about, now a little bit less than 40, because this will open at about a minus 40. So when they get to about a 45 or so, you don't have to know that exact number, another kind of channel opens briefly, and this is called a transient. That's why it's brief, it's called transient, because it's brief. This is called a transient calcium channel, which is also, by the way, voltage gated. So funny channels close, the transient calcium channels open. So we keep depolarizing the cell. When it gets to our minus 40, that's the threshold potential. That's you. So this is our threshold potential. And that opens, instead of voltage gated sodium, which is what we're used to for our neurons in our skeletal muscle cell, this is going to open calcium channels. And these are called fast calcium channels as well as long lasting. So you'll see long lasting or fast and LL for long lasting, so you'll see both of those. We're going to compare that to the contractile calcium channels of the contractile cardiac cells that are slow calcium. Okay, so that's why I like the fast calcium.
When do the fast open?
So they open when the threshold of minus 40 is reached.
But minus 40 is also when transient open?
No, that's the transient's going to open like about a minus 45 or so, minus 50 to 45.
And you said it's 40 positive for the fast then?
So these are the fast funny channels open at minus 60. Threshold is minus 40.
Right.
So just prior to threshold, like a minus 45 to a minus 50, will be when the transient calcium opens. Okay. And then when we get threshold, the fast, long-lasting, voltage-gated calcium channels open. So we have the transient voltage-gated and we have the fast, long-lasting voltage-gated. In contrast to our other types of action potentials, which allow sodium into the cell. This does it with calcium. All right, and then for repolarization, we see voltage-gated potassium. And when that gets back to minus 60, we start all over again, filling the cell up with positive ions. Okay? Now obviously in there is a recovery because we have to get the calcium ions back out and the potassium ions back in. But we don't see that's covered up by the depolarization going on. We don't see a hyperpolarization in that occurrence. So no resting membrane potential, okay? Now remember how I gave you some hands up when we're doing endocrine and reproductive? So for this material, this unit component, I would suggest that you be able to identify, like we first put up on the board, the different potentials, so resting membrane potential, graded potential, and action potential. Threshold potential, what kind of channels are associated with those, as well as, so this would be what kind of a potential? If it's not resting, what would we call this? A graded potential. And so if we're looking at the effect of the sympathetic nervous system, to have more beats per minute, we have to cause this to take place in a shorter period of time. To have a slower heart rate, we have to cause this to take place over a longer period of time. That's a graded potential. Okay, it's gaining positive ions more rapidly, or it's gaining positive ions more slowly. So this is our graded potential. And our autorhythmic cells, that graded potential is due to the funny channels and the transient calcium voltage gated channels. Okay? Then we have an action potential. And both of these are voltage gated. So we have fast calcium voltage gated channels and our potassium voltage gated channels. Okay? So if I have a diagram on your quiz or exam or homework, be able to identify this as the membrane potential changes for autorhythmic cells, and then what channels are involved in the potentials that are created there. Okay? All right, so that's our standard membrane potential changes for autorhythmic cells. Every time the autorhythmic cells reach an action potential, and this would occur at the same time for all the cells forming our sinoatrial node, these positive ions, which are calcium, are going to flow into our contractile cells of the atria, right? So if we go back to this image, when action potential is reached in the autorhythmic cells, we have that spike of positive ions coming into the cell. Some of them are connected to the contractile cells of the atria via gap junctions. And we're going to cause the contractile cells to depolarize, not because of the SA, not because of parasympathetic fibers or sympathetic fibers, but because those ions are coming from the autorhythmic cells. And they're going to enter the first contractile cells via gap junctions. Okay? So when we come back from break, we'll talk about those contractile cells. But first, I want to look at how the autonomic nervous system modifies our heart rate. Okay? So this is talking about the funny current channels. So they're both voltage gated, all right? And they're both going to be activated by the binding of cyclic AMP.
I have a question.
Yeah.
Is this membrane potential changes, is this SA node?
So it's the SA node does it the fastest.
Okay.
If the SA node is damaged, then the AV node will take over.
Okay, because it's faster.
And I don't know if you've ever heard of atrial fibrillation, but it's usually symptomless. People can go for 15, 20 years and they usually have a stroke because of clots forming from the irregular blood flow before they even know that they have atrial fibrillation. So the atria really only just contract briefly just before the ventricles contract. And the ventricle contraction is what's required to move the blood through the body and create that force. So you don't really need atrial contraction for that to occur. So people can live with just the AV node function. All right, so this is really just what I've been going through already. So let's look at the effect. Question number 4. We did 3?
Yes.
Okay. Question number 4. The membrane potential of the region labeled 0 is identified as which of these? The only 2 answers you can guess. You have a 50-50 chance, but I think you know it well enough about that. Although I don't actually have it written on the board, I talked about it. Question 5. The membrane potential labeled 1. So that's before we get to threshold, before we get to the dotted line. So what's causing that region of the graded potential at the site labeled 1? And that answer is on the board. So do we have any normal voltage-gated sodium channels illustrated up there? No. Voltage-gated potassium channels would be creating 3. Funny channels would be creating 4. Voltage-gated fast channels would be creating 0. Okay? That leaves you with your answer for this question. Okay, this does a review that I'm not going to go through. It just kind of talks about the different events. It looks a little overwhelming, but it's everything we talked about is up there. And so what I want to talk about here is the effect of the sympathetic nervous system. All right? Norepinephrine or epinephrine, if it's coming from the adrenal glands, binds to a G-coupled protein receptor. Talked about that with the endocrine system. So would this be an ionotropic or a metabotropic receptor? Is it opening an ion channel or a G protein coupled receptor? Let's just give the answer. So if it's a G protein coupled receptor or anything that's not an ion channel, then it's a metabotropic. Okay? And it can downstream open up ion channels, but if it doesn't open those ion channels directly, it's metabotropic. So the ligand-gated channel for acetylcholine, opening up a sodium ion channel would be an ionotropic, okay? But acetylcholine binding to, say, the smooth muscle of or the nitric acid binding to the receptors in the renal arterioles, that would be metabotropic. All right, so here's our G protein. The alpha subunit with GTP activates adenylate cyclase, and that converts ATP to cyclic AMP. All right? Where did we just talk about cyclic AMP? Binding to the funny channels. Well, now we have a bunch more of the cyclic AMP. So it will open up even more funny channels, okay? And it will open them up earlier. So 2 things happen. More of these open and they open up earlier. What do you mean by earlier? It doesn't have to get all the way down to -60. It'll open up when it's at -50 or -55. That saves time, okay? So if we look at this, this is a -60, and because of norepinephrine and cyclic AMP, it opens up here. It's going to take less time to get to threshold, okay? Therefore, we can have more action potentials within the same period of time. In addition, we activate protein kinase, and that causes more transient calcium channels to open. So what we're doing between these two is adding more positive ions to the cell so it reaches threshold earlier. So we open up more of the transient, we get there faster, all right? So if this is our normal pattern, and here's an action potential resulting in a heartbeat every time the autorhythmic cells get an action potential under the influence of norepinephrine, and we decrease that. We can get a faster heart rate. Okay? I illustrated it last night with the Monday, Wednesday, Friday class. Monday, Wednesday, not Friday. Monday, Wednesday class by saying if you're trying to fill a bathtub, okay, and you're trying to get it to a certain level, you turn on the faucet. And that's the autorhythmic cells adding positive ions via our funny channels and our transient calcium channels, and we reach to that level in standard time. If we open up the faucet even more, and the stream of water coming through the faucet is faster, and we start adding pitchers of water, we're going to get to that level earlier, okay? So that's what the sympathetic nervous system does. It increases the rate at which positive ions enter the cell. Therefore, they move in faster, and we get to threshold more times because we get there more often. So we have a greater number of action potentials in a minute, and that results in a greater number of atrial and ventricular contractions, okay? So this diagram here kind of shows the same thing. I'll go over it with you because it's a little harder to understand than the previous diagram, but here's our beta 1 norepinephrine receptor. In the lungs, we also have norepinephrine, but we have beta 2. So if you use salbutamol, to open up your lungs, your heart rate's going to go up a little bit, but not as much as if it was a beta 1 receptor. Yeah. So you would go, your heart rate would skyrocket every time you use an asthmatic drug instead of just go up a little bit, okay? So pharmaceuticals use the difference in those receptors. So this means a stimulatory G protein, which is our alpha-1 subunit. Our alpha subunit activates adenylate cyclase, which takes ATP, converts it to cyclic AMP. And that cyclic AMP itself will open up our body channels, and because of protein kinase, it's showing cyclic AMP here, but it's because of the protein kinase, will cause transient calcium channels to open earlier, okay? Which is essentially what I wrote here. Now, remember that enzyme phosphodiesterase? What was the role of phosphodiesterase as far as cyclic AMP and cyclic GMP? It inactivated it. So if caffeine blocks the activity of phosphodiesterase, it's like a double negative, which is a positive, which means your heart rate goes up, okay? And your blood pressure can go up as a result of that as well. All right, so autonomic nervous system. So when we talk about the control of heart rate, we'll look at where the sympathetic nerves come from. I don't know if you remember the autonomic nervous system from Bio 430, but in the neck, we have 3 autonomic ganglia along the autonomic chain. We have the inferior, the middle, and the superior, okay? And so the ones coming to the heart are coming primarily from the middle, 2 from the inferior, and those are post-ganglionic fibers that come to the heart. They're not lightning. And so they're just called cardiac accelerator nerves. This is what they do, okay? But we'll look at that when we look at how we regulate heart rate and blood pressure and so on. So, parasympathetic. What's our parasympathetic nerve that's getting to the heart? Splanchnic nerve. We have a splanchnic nerve that's the parasympathetic like we do in the pelvis? Or not? What's it called? It's the wanderer. It's coming through the jugular foramen, down the neck. It's going to innervate the lungs, the stomach, vagus. All right, so vagus nerve comes to the heart, to the SA node, to the AB node. Usually the right vagus nerve does SA node and the left vagus nerve does the AB node, but there can be some overlap. And acetylcholine binds to muscarinic receptors. Okay, you don't have to know which one. There's 5 different ones in the body, primarily, but it binds to muscarinic receptors. So vagus is a preganglionic or a postganglionic? Preganglionic. So actually in the heart wall are postganglionic neurons releasing the acetylcholine that binds to the muscarinic receptors. Because in actuality, one atom lesson here, if this is vagus, would you like my anatomical heart? Then at the SA node is the postganglionic neuron. Both of them are releasing acetylcholine. A few minutes into class, I said here we have ionotropic receptors, and the muscarinic receptors are going to be at the SA node. So the cardiac autorhythmic cells have the muscarinic receptors, and those are all actually getting the acetylcholine from the postganglionic neuron, which is inside the heart wall, not from the vagus. But they're activated because of vagus, okay? And vagus releases acetylcholine to activate the postganglionic neurons. They open up sodium ion channels and then release acetylcholine from the area. So that acetylcholine is actually coming from the postganglionic neurons. Binds to the muscarinic receptor. factors, which activates inhibitory activity. So our alpha subunit is not an excitatory one like we've normally been talking about. Back last semester I said there are some inhibitory actions, so here we see it. It actually inactivates adenylate cyclase. Okay? Norepinephrine and epinephrine activated an excitatory alpha subunit, which turned on adenylate cyclase. This particular alpha subunit inhibits adenylate cyclase, so we get less cyclic EMP. Alright, so fewer funny channels open, and the ones that do open later. So it has to, and instead of opening earlier, they're going to open later. So the resting membrane potential actually gets a little bit lower before we start to open our funny channels. We have more positive ions that we had to add to the cell because the inside of the cell got more negative. And then for the first time now, we're talking about the beta and gamma subunits. Those those open the potassium channels. All right, which way does potassium go? Inside a cell or outside a cell? Outside the cell. So it's like we pulled the plug, pulled the drain on our bathtub. We've turned the spigot down, the faucet handle down, so less water is coming through the faucet, but now we've also opened the plug. So while the tub is filling up with water, it's losing water. It's filling up faster than it's losing, but it's going to take longer, okay? And our transient channels open up later. So this is going to take a little bit longer because we're losing positive ions at the same time we're gaining. And this is going to take longer. So it's going to take us longer to reach our threshold. So the effect of acetylcholine then is that this is our normal heart rate. With acetylcholine, we're going to have fewer action potentials formed because it's going to take us longer to get to that threshold. We're not adding positive ions as rapidly, and at the same time we're adding fewer positive ions, we're losing positive ions. So we don't have that on here because we don't have potassium channels in our normal component, but because of the beta and gamma, then it opens potassium channels.
Maybe these were the beta and gamma came from, I guess.
Our G protein. Let's go back to the, my diagram for acetylcholine is actually another lecture, but if we change this from norepinephrine to acetylcholine, remember we have 3 subunits. We've always talked about the alpha subunit. The other 2 are beta and gamma.
Okay.
So the alpha subunit for acetylcholine is not excitatory, it's inhibitory, so it blocks this from happening, okay? And then the beta and gamma subunits open up potassium. And because potassium goes out of the cell, we lose positive ions when we're trying to gain them. So while we don't need the sympathetic nerves, and we don't need the parasympathetic nerves to cause our autorhythmic cells to depolarize, we use them to modify the rate at which they depolarize. So we use them to depolarize faster if we're using the sympathetic nerves, or depolarize more slowly if we're using parasympathetic. Okay? So in the PhysioX activity, which should be ready tomorrow, I'm still writing it, you are going to be using the PhysioX. So before we go on break, let me just pull that up. Did anybody in here not use PhysioX in the past? Okay, a few people. Couple of ways to access it. If you have a new textbook,